Hello my dear friends, welcome to S Class Education. So today we are going to see exercise of chapter number 3, 12th standard biology, inheritance and variation. Let us start. Question number 1, multiple choice question. Sub question 1, F2 phenotypic ratio of incomplete dominance in Mirabilis chalapa is option A 2:1:1, option B 1:2:1, option C 3:1, option D 2:2. So correct answer is option B 1:2:1. Second question, in dihybrid cross F2 generation offsprings show 4 different phenotypes while the genotypes are A6, B9, C8, D16. So the correct answer is option B 9. Sub question 3, a cross between an individual with unknown genotype for a trait with recessive plant for that trait is option A back cross, option B reciprocal cross, option C monohybrid cross, option D test cross. So correct answer is option D, that is test cross. Question 4, when F2 phenotypic and genotypic ratios are the same, then it is example of option A incomplete dominance, option B complete dominance, option 3 multiple alleles, and option D cytoplasmic inheritance. So correct answer is option A, that is incomplete dominance. Next question is, if the centromere is situated near the end of the chromosome is called option A metacentric, option B acrocentric, option C submetacentric, and option D telocentric. So correct answer is option B, that is acrocentric. Question 6, chromosomal theory of inheritance was proposed by option A Sutton and Boveri, option B Watson and Crick, option C Miller and Urey, option D Oparin and Haldane. So the correct answer is option A, that is Sutton and Boveri. Question number 7, if the genes are located in a chromosome as P, Q, R, S, T, which of the following gene pairs will have least probability of being inherited together? Option A P and Q, option B R and S, option C S and T, and option D P and S. So correct answer is option D, that is P and S. Find the mismatch pair. A is Down syndrome 44 plus XY, B is Turner syndrome 44 plus XO, C is Klinefelter syndrome 44 plus XXY, and D is normal female 44 plus XXX. So correct answer is option A. Correct answer is option A, that is Down syndrome is equal to 44 plus XY. Next question is question number 9. A colorblind man carries a woman who is homogeneous for normal color vision. The probability of their son being colorblind is 0%. Option A is the correct answer. Question number 2, very short answer type questions. One is explain the statements. A, test cross is back cross, but back cross is not necessarily a test cross. So the answer is, in back cross, if F1 generation is crossed with a recessive parent, it will be a test cross. But if F1 generation is crossed with a dominant parent, it will be not a test cross. And therefore, the test cross is a back cross, but the back cross is not a test cross. Next question, law of dominance is not universal. Answer, the law of dominance is not universal because in certain cases, such as in the case of incomplete dominance, one allele shows incomplete dominance over the recessive allele, and in codominance both the alleles are expressed. Next question, define the following terms. First term is dihybrid cross. A cross between parent differing in two heritable traits is called as dihybrid cross. For example, cross of pure tall round seeded plant with dwarf wrinkled seeded plant. Second is homogeneous. An individual possessing identical alleles for a particular trait is called as homogeneous or pure for that trait. C is heterogeneous. An individual possessing contrasting alleles for a particular trait is called heterogeneous. Heterogeneous does not breed true for that trait and produces two types of gametes, for example F1 generation hybrids. D is test cross. It is a cross of F1 progeny with homogeneous recessive parent, and it is used to test the homogeneous or heterogeneous nature of dihybrid. It is kind of back cross. Okay, so next is what is allelosome. If you want, you can take screenshot simultaneously. So please take out the screenshot. Okay, done. Next is what is allelosome. So if you want, please take out the screenshot simultaneously. The chromosomes which are responsible for determination of sex are known as sex chromosome or allelosome. What is crossing over? Crossing over is a process that produces new combinations of genes by interchanging and exchanging of corresponding segments between non-sister chromatids of homologous chromosomes. Give one example of autosomal recessive disorder. An example is sickle cell anemia. Sixth question, what are X-linked genes? Answer, genes which are located on non-homologous region of X chromosome. So here by mistake I have written answer, so just ignore. Of X chromosome are called as X-linked genes. What are halondric traits? Genes located on non-homologous region of Y chromosome are called as Y-linked genes or halondric genes. The traits that are determined by Y-linked and gene are called as Y-linked traits or halondric traits. Next is give an example of chromosomal disorder caused due to non-disjunction of autosomes. Turner syndrome, that is X monosomy or XO female, is a sex chromosomal disorder caused due to non-disjunction of chromosomes during gamete formation. Next is question number 9. Give one example of complete sex linkage. Example of X-linked trait is hemophilia. Short answer type question. Enlist 7 traits of pea plant selected or studied by Mendel. 7 pairs of contrasting visible characters in pea plant or Pisum sativum is as follows. So in this question, if you want, you can draw the diagrams, or even if you don't draw, it won't matter. So just you have to draw a chart in which you can make 2 columns of dominant and recessive, and then you can make rows containing seed shape. Dominant, under dominant you can write round, under recessive you can write wrinkled, and then for the seed color another row you can write yellow in the dominant column and green in the recessive column. Like this also you can do. Okay, so next question, why law of segregation is also called the law of purity of gametes? The gametes which are formed after segregation are always pure for a particular trait. A gamete may carry either dominant or recessive factor but not both. That's why it is also called as law of purity of gametes. Write a note on pleiotropy. So when a single gene controls 2 different traits, it is called pleiotropic gene, and the phenomena is called as pleiotropy or pleiotropism. What are the reasons of Mendel's success? The reason of Mendel's success are his experiments were carefully planned and involved large sample. He carefully recorded the number of plants of each type and expressed his results as ratios. In the pea plant, contrasting characters can be easily recognized. The 7 different characters in pea plant were controlled by a single factor. The factors are located on separate chromosomes, and these factors are transmitted from generation to generation. He introduced the concept of dominance and recessiveness. If you want, you can take the screenshot. Please increase the video quality so that you may be able to get clear screenshots. Next question is question number 5. Father is responsible for determination of sex of child and not the mother. Justify. So answer, that is A and S. So maybe A is missing here. Just write it. So if a sperm containing X chromosome fertilizes egg, the diploid zygote is formed that grows into a female child. If a sperm containing Y chromosome fertilizes the egg, then diploid zygote is formed that grows into a male child. This indicates that the sex of a child depends on the type of sperm fertilizing the egg, and hence the father is responsible for determination of sex of child and not the mother. What is linkage and how many linkage groups do occur in human being? The tendency of 2 or more genes present on the same chromosome that are inherited together is known as linkage. Humans have 23 linkage groups because they have 23 pairs of chromosomes. Write a note on PKU. Answer: it is an inborn metabolic disorder caused due to recessive autosomal genes. When recessive genes are present in homozygous condition, phenylalanine hydroxylase enzyme is not produced. So phenylalanine A, that is the hydroxylase enzyme, is not produced. This enzyme is essential for conversion of amino acid phenylalanine into tyrosine. Due to absence of this enzyme, phenylalanine is not converted into tyrosine, and hence phenylalanine and its derivatives are accumulated in blood and cerebrospinal fluid, that is CSF. It affects development of brain and causes mental retardation. Excess phenylalanine is excreted in urine, and hence this disease is called as phenylketonuria. So just check out the answer, or if you want you can click out this, take out the screenshots. Okay, done. Next question. Compare X chromosome and Y chromosome. You can make columns and write like difference between, or also you can follow this paragraph method. So answer: X chromosome is straight, rod-like and longer than Y chromosome. Next is Y chromosome is acrocentric, and similarly X chromosome is metacentric. X chromosome has large amount of euchromatin and small amount of heterochromatin. And if we check out the for Y chromosome, okay, so euchromatin has large amount of DNA material and hence genetically active, and for Y chromosome it has small amount of euchromatin and large amount of heterochromatin, and hence it is genetically less active or inert. Both X and Y chromosomes show homologous and non-homologous regions. Homologous regions show similar genes, while non-homologous regions show dissimilar genes. X-linked genes are present on non-homologous region of X chromosome, while Y-linked genes are present on non-homologous region of Y chromosome. Okay, done. Next question. Explain the chromosomal theory of inheritance. The chromosomal theory of inheritance states that the chromosomes are present in pairs in somatic cells. During gamete formation, homologous chromosomes pair, segregate and assort independently during meiosis. Thus, each gamete contains only one chromosome from a pair. Nucleus of gametes contain chromosomes which carry all hereditary traits. Male and female gametes carry all the hereditary traits. They are the link between parents and offspring. The fusion of haploid male gamete and haploid female gamete restores the diploid number of chromosome of the species. So next question is question number 10, in which we have given a pedigree. We have to observe the pedigree, and then depending upon the pedigree chart we have to answer the questions. Okay, so let's check out what are the questions and what will be the answers. First is identify whether the trait is sex linked or autosomal. So the given trait is sex linked. Give an example of a trait in human being which shows such pattern of inheritance. So answer is hemophilia. Next is question number 4, in which we have to match the columns, column 1 with column 2. So column 1, 21 trisomy, the condition is Down syndrome. X monosomy, the condition is Turner syndrome. Holendrick traits, the condition is hypertrichosis. Feminized male, the condition is Klinefelter syndrome. You can check out the answers which are given here. Okay. Next. Long answer type questions. What is dihybrid cross? Explain it with suitable example and checkerboard method. So we have to answer like long answer we have to give. So dihybrid cross, a cross between parents differing in two heritable trait is called as dihybrid cross. For example, cross of pure tall round seeded as well as plant with dwarf wrinkled seeded plant. Mendel also performed the dihybrid cross between pea plants that differed in two pairing of contrasting characters. So for example, when we cross a true breeding pea plant bearing round and yellow seed with true breeding pea plant bearing wrinkled and green seeds, if we cross, then we get pea plant bearing round and yellow seeds in the F1 generation. And when the F1 generation are selfed, we get a ratio of 9:3:3:1 in the F2 condition or F2 generation. where 9 plants bear yellow round seeds, 3 plants bear yellow wrinkled seeds, 3 plants bear green round seeds, and 1 plant bears green wrinkled seeds. So please check out the chart also, so that you will get the idea whatever we just read, whatever I just read, because just by reading you won't get whatever is written there. So just check out the chart so that you will get the idea. Okay, done. So I think we should move to the next question. Explain with the suitable examples. We have to explain the independent assortment. Okay. This law is based on dihybrid cross. So they have already mentioned that this law is based on the dihybrid cross. Okay. So next is, it is basic principle of genetics developed by a Mendel. It describes how different genes or alleles present on separate chromosomes independently separate from each other. So these alleles are then during formation of gametes, and these alleles are then randomly united in fertilization. In dihybrid cross, F2 phenotypic ratio, that is 9:3:3:1, indicates that the two pairs of characters behave independently of each other. It can be concluded that the two characters under consideration are assorted independently, giving rise to different combination. Statement of law of independent assortment. The law states that when dihybrid possessing two pairs of contrasting factors forms gametes, the factors in each pair segregate independently of the other pair. So again the same chart is given here because the question is same. If you want, just take out the screenshot and then we have to move towards the next. Next is define test cross and explain its significance. So what is test cross? The cross of F1 hybrid with the homozygous recessive parent is known as a test cross. It is used to test whether the individual is homozygous, that is pure homozygous, or heterozygous, which is hybrid. Test cross is easy, simple, repeatable and predictable. Test cross can be used to find out genotype of any plant with dominant expression, but it is not known whether it is homozygous or heterozygous for that trait. For example, a pea plant having violet flowers is crossed with a pea plant with white flowers. If all flowers produced are violet, we can conclude that plant is pure or homozygous, and if we get violet and white flowers in 1:1 ratio, then we can conclude that the plant is heterozygous. So in this way, test cross is also used to introduce useful recessive traits in hybrids of self-pollinated plants during rapid crop improvement programs. So if you want, you just have to take out screenshot, please, or you can also check out this into your textbook. Next one is, what is parthenogenesis? Explain the haplodiploid method of sex determination in honeybee. So parthenogenesis is the development of embryo directly from egg cell or a male gamete. It is kind of apogamy. So in honeybees, chromosomal mechanism of sex determination is haplodiploid type. In this type, sex of individual is determined by the number of set of chromosome received. Females are diploid, that is 2, n is equal to 32, and males are haploid, so that n is equal to 16. The female produces haploid eggs, that is n is equal to 16 by meiosis, and male produces haploid sperm, that is n is equal to 16 by mitosis. If the egg is fertilized by sperm, the zygote develops into a diploid female, that is 2n is equal to 32, queen and worker, and unfertilized egg develops into haploid male, so by way of parthenogenesis. A diploid female gets differentiated into either worker or queen depending upon the food they consume during their development. Diploid larva which get royal jelly as food develops into queen, and other develops into workers. So here you can just check out the chart and you have to make the chart. Next is question number 5. In the answer for inheritance of X-linked genes, Madhav had shown carrier male. So his answer was marked incorrect. Madhav was wondering why his marks were cut. Explain the reason. Males can never be carriers. They have single X and other Y chromosome. In X-linked inheritance, the genes are present on the non-homologous region of the X chromosome. Males do not have other X, and hence if the genes are present on his X chromosome, they will not be suppressed in them. The Y chromosome does not have dominant gene to hide this expression. as there is no homologous region 2, but in case of females there are double X chromosomes, and hence if X-linked gene is recessive, the other X can hide the expression of such X-linked gene. Thus she becomes a carrier without showing any physical characters. She is physically normal and does not suffer from such X-linked recessive disorder. Thus Madhav will get his answer wrong due to incorrect concept. Next is, with the help of neat label diagram, describe the structure of chromosome. So structure of chromosome. Chromosomes are best visible under microscope when the cell is at metaphase stage. It is because at this stage chromosomes are highly condensed. Typical chromosome consists of two chromatids joined together at centromere or primary constriction. Primary constriction consists of a disc-shaped plate called as kinetochore. It is at the kinetochore spindle fiber get attached during cell division. Besides primary constriction, some few chromosome possesses additional one or two constrictions called as secondary constriction. At secondary constriction 1, nucleolus becomes organized during interphase. A satellite body is attached at secondary constriction 2 in very few chromosomes. Each chromatid in turn contains a long unbranched slender, highly coiled DNA thread called as chromonema, extending through the length of chromatid. Chromatid consists a double-stranded DNA molecule which extends from one end to the chromosome of other end of the chromosome. So this is the diagram of chromosome. Next question is, what is criss-cross inheritance? Explain with suitable example. Where is its answer? Okay, so here is the answer. Criss-cross inheritance is a type of inheritance in which the genes are passed on from father to daughter and then to her son. That is from father to daughter to son. Okay. Next is, that is from male to female and from female to male. That is grandson. In other words, it is also said that the transmission is from the grandfather to the grandson through his daughter. Inheritance of color blindness shows criss-cross pattern. So it is X-linked recessive disorder where person is unable to distinguish between red and green colors as both the colors appear grey. It is caused due to recessive X-linked which prevents formation of color sensitive cells, the cones in the retina of eye. The homozygous recessive female and homozygous recessive male, it is homozygous, okay, are unable to, it is heterozygous or hemizygous, are unable to distinguish between red and green colors. The frequency of colorblind woman is much less than colorblind male. Dominant X-linked gene is necessary for formation of color sensitive cells in the retina of eye. So just check out the charts. You have to write this point also as we have to mention the condition. So just check out the points. Done. Describe different types of chromosomes. Depending upon the position of centromere, chromosomes are divided into following types. First is metacentric. So in metacentric chromosome, the centromere is situated in the middle of the chromosome. The two arms are of equal length. Submetacentric, here the centromere is situated some distance away from the middle and one arm is shorter than the other, like L. Next is acrocentric. In acrocentric, the centromere is situated near the end of the chromosome, so one arm is too short and it appears like J shape. Telocentric. In telocentric, the centromere is situated at the tip of the chromosome and one arm is like it represents only like one arm and like rod shaped. So you can check out the diagrams also. Based on the functions, they are divided or they are classified into two types. So autosomes, those which decide the body character, and allosomes, those which decide the sex of the individual. So in this way we have completed the exercise of this chapter. I hope you liked this video. If you liked, please share it with your friends and also subscribe our channel so that you will get notified whenever I upload the new videos. So thank you so much for watching this video and bye bye.